friends, I am Romila Lemos, Assistant Professor from the Department of Forensic Biology, Institute of Forensic Science, Mumbai. Today's topic of discussion and learning is Forensic Serology Part 11. I will be presenting this topic on behalf of myself and Ms. Deepika Bhandari. The following modules will be covered for this lecture. Module 1, Introduction to other biological fluids. Module 2, Properties of vaginal secretions, menstrual blood, urine, feces and vomit. Module 3, Collection, preservation and dispatch of specimen. Module 4, Preliminary examination of vaginal secretion and menstrual blood. Module 5, Preliminary examination of urine, feces and vomit. Module 6, Confirmatory examination of vaginal secretion and menstrual blood. Module 7, Confirmatory examination of urine, feces and vomit. Module 8, Conclusion. Besides blood, saliva and semen, a number of other biological fluids play an important role as a part of forensic serology. The identification of vaginal secretions and menstrual blood is important for the investigation of sexual assault cases. Such identification can help to corroborate allegations of sexual assault. If vaginal secretions are found in the stain on a suspect's clothing, the evidence has probative value to corroborate an allegation of a sexual act. Additionally, post-coital drainage stains on clothing or bedding are often recovered at crime scenes. These stains usually consist of a mixture of semen and vaginal secretions. In such cases, the presence of vaginal secretions in these stains indicates the occurrence of sexual intercourse. Sometimes vaginal secretions can be transferred onto a perpetrator during a sexual assault. For example, the presence of vaginal secretions on a suspect's genital area can indicate the occurrence of sexual intercourse. Furthermore, the presence of vaginal secretions on an object can corroborate an allegation of vaginal rape with a foreign object. If a sexual assault victim is in menses when an assault occurred, blood evidence such as the victim's bloodstains located on the suspect's clothing may be recovered at the scene. In this case, the identification of menstrual blood would corroborate an alleged rape. Therefore, it is necessary to distinguish between peripheral and menstrual blood in investigations of sexual assault where blood evidence is found at the scene. The identification of the presence of urine is useful for the forensic investigation of an alleged sexual assault and harassment involving urination. More importantly, the identification of urine stains can aid in the investigation of homicides involving either ligature or manual strangulation. In these incidents, strangulation victims often involuntarily excrete urine prior to death. The locations of urine stains at the crime scene provide useful information to determine the site where the violence has occurred. The examination of feces has been used in criminal investigations for over a century. One of the aspects of fecal analysis is to determine a common origin of the reference sample and the fecal evidence, thus potentially linking a suspect to a crime scene. The presence of fecal matter may corroborate a sexual assault involving sodomy, assault with fecal matter, vandalism and burglary during which the perpetrator defecated at the scene. A normal human vagina is covered by the squamous mucosa, which is composed of stratified squamous epithelial tissue. Lying under the squamous mucosa is the submucosa, which contains an abundance of connective tissue and capillaries. Below the submucosa, is the muscularis, which is made up of smooth muscle. The squamous mucosa consists of multiple layers of cells. As these epithelial cells reach the apical layer, the superficial layer, the cells are truly differentiated with small and dense nuclei. Vaginal epithelial cells do not accumulate keratin, which is different from skin cells. The intermediate and superficial layer cells contain abundant glycogen in their cytoplasm. The presence of glycogen in these cells is an indication of normal development and the differentiation of the vaginal epithelial cells. The apical surface of the vaginal squamous mucosa is usually covered by mucus that is secreted from glands that are located deep in the epithelium. Menstruation is the periodic discharge of blood and the elimination of the degenerated lining of the endometrium from the uterus of a non-pregnant woman. From menarche to menopause, women may menstruate up to 400 times during their reproductive age. During the uterine cycle, repetitive physiological changes occur in the functionalities of the uterine endometrium. The cycle is divided into three phases: the menstrual, proliferative and the secretory phase. During the menstrual phase, the functionalities degenerates and is sloughed off from the uterine wall and bleeding occurs, known as the menses. During the proliferative phase, the functionalis begins regeneration in which the spiral arteries are proliferated. During the secretory phase, the spiral arteries are further developed and coiled. Menstrual fluid contains blood, the functionalis, layer tissues and mucus. The urine is drained from the kidneys through the ureters and is stored in the bladder before it is finally excreted through the urethra. Urine is an aqueous solution consisting largely of water. Urea is the most abundant waste product in urine, resulting from the elimination of ammonia that is produced from the metabolic process of amino acids. The average urea concentration in human urine is approximately 9 grams per liter. Other major components are creatinine, uric acid and ions such as phosphate, sulfate, chloride, sodium and potassium. Healthy individuals also excrete a small amount of protein. Urine has a characteristic yellow color. Subsequent to excretion from the body, urine acquires an odor that results from the ammonia that is released from the breakdown of urea by bacteria. Feces are a type of waste matter that is the direct result of food that has been processed by the digestive system. Human feces contain undigested foodstuffs, sloughed intestinal epithelial cells, intestinal bacteria, bile pigments, electrolytes and water. Feces are formed in the intestines during the last phase of digestion. Feces first enter the colon in liquid form. Most of the nutrients are absorbed on the surface area of the small intestine. In the large intestine, water, sodium and chloride are absorbed on the surface of the lumen. The remaining luminal contents are converted to feces. Vomiting is the forceful expulsion of the contents of the stomach through the mouth. It is usually preceded by salivation, sweating and the sensation of nausea. Vomiting can be caused by a wide variety of pathological conditions. Vomiting may be a specific response to acute intoxication in homicidal poisoning cases, or it may be caused by trauma in a violent assault. Thus, the forensic identification of the gastric fluid can corroborate a criminal act. The vomitus consists of ingested food particles from the stomach, hydrochloric acid, mucus, hormones such as gastrin, a number of enzymes including lipase, gelatinase, pepsinogens, etc. The liquid stains found at the crime scene should be collected with a clean, sterile syringe or disposable pipette and transferred to a clean, sterile test tube. Solid particles, in case of feces and vomitus, can be transferred to a clean test tube with a clean spatula. A clean cotton cloth can be used to soak up liquid sample. Wet fluid samples, if they are collected, must be kept in a refrigerator. These specimens should be submitted to the laboratory as soon as possible. The specimen should be labeled with case number, item number, date, time, location and evidence collector's name. Small objects bearing wet fluid stains should be allowed to air dry, then collected as is. Stains on large objects that cannot be removed from a crime scene should be transferred onto clean cotton cloth allowed to air dry before packaging in a paper container. Each object and container must be properly labeled. Dried fluid stains on weapons, garments and other movable objects should be collected separately by collecting the entire item. Each item should be placed in its own paper container and these should be sealed and labeled properly. If the stain is located on an object, dried fluid stains on weapons, garments and other movable objects should be collected separately by collecting the entire item. Each item should be placed in its own paper container and these should be sealed and labeled properly. If the stain is located on an object that can be cut, then a portion of the item containing the blood stain can be removed by cutting with a clean, sharp instrument. Each cutting should be packaged separately and labeled accordingly. An unstained portion of the item should be collected and packaged as a control. Let us look at the identification of vaginal secretion by Lugol's iodine staining and periodic acid shift method. The technique is based on the principle that iodine reacts with intracellular glycogen to exhibit a color. Lugol's stock solution in an aqueous solution of iodine and potassium iodide. Iodine atoms fit into the helices of glycogen to form a dark brown glycogen iodine complex. Vaginal epithelial cells can also be stained using the periodic acid skiff method. The cytoplasm of the vaginal epithelial cells is stained magenta and the nucleus is stained purple. The next technique is the Dain staining technique. The Dain staining method has been developed to distinguish between different types of cells. In this assay, skin cells are stained red and orange, buccal cells are stained predominantly orange-pink with red nuclei, and the vaginal cells are stained bright orange with orange nuclei. Let us look at the identification of vaginal acid phosphatase. A number of different acid phosphatase isoenzymes have been identified in the human tissue. Historically, vaginal acid phosphatase has been used as a biomarker for the identification of vaginal secretions using acid phosphatase catalytic assays. In sexual assault investigations, it is important to distinguish between vaginal acid phosphatase from the prostate acid phosphatase originating from semen exposure. These two enzymes have identical molecular weights, enzymatic specificities and responses to the same inhibitors. Nevertheless, vaginal and prostate acid phosphatases can be distinguished using agarose gel electrophoresis. The identification of vaginal bacteria. Lactobacillus taxa are the predominant bacteria in the vagina of women of reproductive age, and they play an important role in protecting the host against invasive pathogenic organisms. The most frequently occurring lactobacillus species found in the vagina are L. iners, L. crispatus, L. gasseri and L. jensenji. Lactobacillus can be observed based on the sequences of DNA markers such as the 16S rRNA gene and the intergenic spacer region between the 16S rRNA and 23S rRNA genes. However, Lactobacillus taxa may also be present in semen and in female urine. The D-dimer assay is used for the analysis of menstrual blood. During fibrinolysis of the menstrual cycle, cross-linked fibrin is cleaved by plasmin, producing a degradation product known as D-dimer. Assays for D-dimer fragments have been utilized in the forensic identification of menstrual blood. A number of different formats of D-dimer assays can be used, such as enzyme-linked immunosorbent assay, immunochromatographic assays and latex agglutination assays. The lactate dehydrogenase assay may also be used for the detection of menstrual blood. LDHs are found in various human tissues. 5 isoenzymes can be found in blood: LDH1, LDH2, LDH3, LDH4 and LDH5. The 5 isoenzymes can be separated using electrophoresis and detected using a colorimetric assay. In the peripheral blood, LDH1, 2 and 3 are predominant, while LDH4 and 5 are predominant in the menstrual blood. Thus, menstrual blood can be distinguished from the peripheral blood. Let us look at the preliminary tests for urine. In forensic identification, urine stains can be located by visual examination based on the characteristic yellow color of urine and the direction of the distinctive odor of urine stains. Under alternative light sources, urine stains emit a fluorescent light that facilitates the locating of urine stains in clothing and bedding. Chemical analysis can be carried out to detect the major inorganic anions in urine such as phosphate and sulfate, as well as the major organic compounds in urine such as urea, creatinine and uric acid. However, these assays are not specific to urine. Let us look at the identification of urea. The para-dimethyl aminocinnamaldehyde, that is the DMAC assay, is simple and rapid and is the most commonly used presumptive assay for the forensic identification of urine stains. The DMAC assay can be performed using two different methods, that is the colorimetric and the fluorometric methods. In the colorimetric method, a portion of a stain is cut and extracted with distilled water. The extraction is transferred onto a piece of filter paper and is allowed to dry. A drop of 0.1% DMAC solution is then added to the filter paper, wherein DMAC reacts specifically with urea if present, producing a pink colored or magenta colored product. DMAC does not react with creatinine, ammonia or uric acid. The fluorometric method is useful for locating urine stains on large pieces of evidence such as clothing and bedding and to detect the patterns of urine stains. In the fluorometric method, the evidence to be examined, such as the garment, is covered by a sheet of filter paper that has been pre-absorbed with the DMAC solution. The evidence and the filter are then wrapped together in a sheet of aluminum foil and are left overnight in a press. Using a light source at 473 to 548 nanometers, the DMAC treated urine stain fluoresces. The fluoresced stain is best observed with a 549 nanometer filter. In addition to the DMAC test, the identification of urea that is present in urine stains can be carried out by urease assays. Ureases catalyze the breakdown of urea, thus releasing ammonia and carbon dioxide. The ammonia is detected using an acid-base indicator bromothymol blue, which exhibits a blue color, or by manganese or silver nitrates, which exhibit a black color. Let's see how the identification of creatinine is taken place. Creatinine is produced during normal muscle cell metabolism. The amount of creatinine excreted in urine is proportional to the muscle mass of an individual. The creatinine present in urine may be detected using the Jaffe color test. In this test, picric acid is used to convert creatinine under alkaline conditions to form creatinine picrate, which is a bright red product. Let's look at the identification of fecal matter. A fecal analysis includes macroscopic and microscopic examination, chemical test and fecal bacterial identification. The macroscopic and microscopic examination. The color and odor of human feces are useful characteristics for fecal identification. When fecal stains are analyzed, the microscopic examination of the feces can be performed on an aliquot of fecal suspension. The presence of characteristic undigested foodstuffs can indicate human feces. Urobilinoids are generated from the degradation of heme and are excreted into feces. These compounds are brown colored and are responsible for the characteristic color of the feces. The urobilinoids can be detected using the Schlesinger and the Edelman tests. In the Schlesinger test, a sample is mixed with saturated zinc acetate in ethanol solution to form a urobilinoid zinc chelation complex that emits a characteristic green fluorescence under the ultraviolet light. The Edelman test is a variation of the Schlesinger test. A sample is treated with a mercuric salt solution to yield a pink colored compound. Further treatment with a zinc salt produces fluorescence. Let's see the fecal bacterial identification. The human intestinal microbiota contains more than 4,000 bacterial species. Bacteroids accounts for approximately 30% of fecal microbiota that are the prominent bacteria in the human feces. The identification of bacteroids can be carried out by detecting specific DNA sequences of the RPOB gene, which encodes the beta subunit of bacterial RNA polymerase. Two fecal predominant bacteroid species, that is B. uniformis and B. vulgatus, can be detected in feces. The identification of vomitus. The color of vomitus may be of forensic interest. Fresh blood in the vomit is usually bright red and suggests bleeding due to injuries, while dark red blood clots suggest bleeding in the stomach due to pathological conditions such as an ulcer. A microscopic examination helps identify recently ingested food particles that are present in the sample. The presence of gastric fluid in vomitus can be identified by the detection of pepsins secreted from the stomach. This identification test is based on the proteolytic activity of pepsins. In the pepsin proteolytic assay, fibrin blue is used as a substrate for pepsin, that is colorless. In the presence of vomitus, pepsin cleaves fibrin blue and releases a chromophore that is now soluble in water and therefore exhibits a blue color. However, this method cannot distinguish the vomitus of humans from that of other vertebrates. Non-destructive confirmatory identification methods such as fluorescence spectroscopy and Raman spectroscopy can potentially be useful for the identification of vaginal secretions. Recently, the analysis of tissue-specific gene expression has been utilized for the identification of vaginal secretions. Using RT-PCR technique, the mRNAs of the tissue-specific genes of vaginal epithelial cells can be detected. Two commonly used markers for vaginal secretion identification are MUC4 and HBD1. Apart from this, RNA-based assays may also be used. Matrix metalloproteinase, that is MMP genes, are considered tissue-specific markers for human endometrium tissues. MMP7 and MMP11 mRNA expressions are elevated at the menstrual phase and remain at high levels during the proliferative phase. Using the RT-PCR technique, MMP11 mRNA and MMP7 mRNA can be detected in menstrual blood from the 1st to the 8th day of menstruation. Let us look at the confirmative assay for urine. TAM horsefall protein or THP, also known as uromodulin, is the most abundant protein in urine and accounts for 40% of the urine proteins. THP is a urine-specific biomarker for forensic urine identification. It can be detected using an enzyme-linked immunosorbent assay, that is ELISA, or an immunochromatographic assay. RSID urine, which utilizes a polyclonal rabbit antibody that is specific to THP. This test is rapid and simple and thus can be used as a screening test in laboratories and as a field test at crime scenes to determine the presence of urine. Besides, derivatives of 17 ketosteroids are excreted into the urine to be eliminated from the body. These 17 ketosteroid conjugates can be identified using liquid chromatography mass spectrometry or LCMS. Identification of the body fluids thus shows a wide forensic application and can be effective as corroborative evidence in a number of cases. As such, proper identification and characterization of these fluids is very much essential. Thus, we have come to the end of the chapter on the various preliminary and confirmatory assays used for the testing of biological fluids. It is important to understand their forensic significance, and I hope that you have learned a lot in this lecture. I will see you again soon in the following episodes with more information and study lessons. Till then, keep learning. Thank you.